Good morning students, I am Rajiv Shen and today I am going to teach you the DNA transcription. So, and this is one of the most important thing. In fact, I am not going to teach you transcription, rather correctly I would like to say that I will teach you the differences between prokaryotic and eukaryotic transcription. Difference between prokaryotic and eukaryotic transcription. Although this is not a big topic, but this is one of the most important topic because even in CBSE examination as well as for NEET preparation, there are so many questions which have been framed in this particular topic. So I would like to teach you this particular part. Now, before going into the difference between the prokaryotic and eukaryotic transcription, let us understand, many of you must have known this, what is the definition of transcription, rather what is actually the transcription process. So basically, as you know, most of you must be knowing, that transcription is the process of copying the genetic information from template DNA to RNA. It is the copying of genetic information from template DNA to RNA. Copying of genetic information from template DNA to RNA. Here in this particular definition, I have already elaborated that which of the two strands of the DNA is basically utilized in the process of transcription, which is the template strand. Because you must have gone through that DNA replication. In DNA replication, both the strands of the DNA is involved in the process of replication. In DNA replication, both the strands are involved in the process of DNA replication, but in transcription, only one of the strands. Here in this particular session, I am not going to elaborate to the process of entire transcription, because as you know, the process of transcription is basically divided into three primary processes. The first process is the initiation, then the elongation, and last is the termination. This, in my next video, I will elaborate you separately prokaryotic as well as eukaryotic transcription, initiation, transcription, that means prokaryotic transcription, initiation, prokaryotic transcription, elongation, and prokaryotic transcription, termination. Although ultimately into this depth it is not given in their NCERT book, but you must know all these things. Okay. Now here I would like to define you that basic difference between prokaryotic and eukaryotic transcription. As you know, if I say like this, prokaryotic cell, eukaryotic cell. In prokaryotic cell, basically, as you know, the process of transcription, that is from DNA to RNA, is taking place inside the cytoplasm, that is the cytosol. If I draw a cell of prokaryotic over here, what is there? Suppose this is the DNA strand. It is there in the cytoplasm of the cytosol. It is naked DNA. As you know, although I am drawing it in single linear way, but actually, as you all know, that is not linear. There is a circular DNA which is present, single circular double-stranded DNA, but I am writing like that for your better understanding. From DNA to mRNA, and then to taking place inside the cell, cytoplasm, not inside the nucleus. But as you know, in eukaryotic cell there is a well-defined nucleus. In prokaryotic cell there is no well-defined nucleus. As you know, in spite of nucleus there is a terminology that is nucleoid. The genome of the prokaryotic cell we used to say is a nucleoid or the genophore or incipient nucleus. These are the various terminologies which has been used generally in different books you will find over there. But here you will find that one nucleus is there. Nucleus is there. As you know, nucleus is double bond structure. And here, this DNA is situated. Here this DNA is situated and from DNA mRNA is formed inside the nucleus. This part only, that is the transcription process or the replication, even the replication of the DNA is also taking place. Even the modification of RNA is also taking place inside the nucleus. This is the eukaryotic cell. Okay. Now, so this is the one major difference. The first difference is that transcription process is taking place in the cytosol here in case of prokaryotic cell. In eukaryotic cell, it is inside the nucleus. As you know, DNA is the so this is transcription. Formation of RNA, although in this particular most of the time it is the mRNA which has been transcribed from DNA. It is the DNA. The mRNA is formed, and it is the mRNA which basically in prokaryotic it is the cytoplasm, that is cytosol, and eukaryotic it is the nucleus. Where this transcription process is formed. Now I am coming to this. This is the first difference. Second difference is that here the mRNA which has been formed, it is a complete structure. There is no any junk element which has been found in the mRNA. Junk element means there is no any such type of introns is there, which is used to be the junk element, is not in the mRNA. But here in prokaryotic cell, it is the complete mRNA, mature mRNA. But here in this case, the mRNA which has been formed is not the mature mRNA. Basically, this mRNA contains certain junk elements, like that I would like to describe in a different way. The mRNA which has been formed over here is like that. It's something like that. Like right here is one part, this is another part, one part, another part. So what you see, this mRNA contains certain black, then red, then black, then red. I am demarcating it because this is the introns, this is the exons. Again introns, INT and exons, yes, and introns. So this is introns, exon sequences is there, which is and this introns is basically the intervening sequences, and exons is the basically the expressed sequences. Express sequence contains the genes basically which manufacture the proteins. The protein manufacturing unit is the exons, and introns are the intervening units. So this mRNA is not the mature mRNA. It cannot form the proteins. For that there will need to be cleaved out. That means it has to be cleaved over here, and here, and ultimately this mRNA is formed. And this process is known as splicing. This process is called splicing, and splicing is the characteristic feature of this. I mean to say this process is going on inside the nucleus. You can say this is a post-transcriptional process. You can say this is a post-transcriptional process where introns are cleaved down and exons are added to the one specific type of enzyme is that is ligase enzyme, ligase, which basically cleaved that portion of the exons. Okay, so this is one thing. One more thing is that is, suppose this is 5 prime and this is the 3 prime. As you know that it is formation is from 5 prime to 3 prime direction. From the directionality point of view, it is from 5 prime to 3 prime. Now there is a capping process is done. 5 prime capping. This is the another important additional feature which has been done in this particular case of eukaryotic cell. 5 prime capping. 5 prime capping. 5 prime capping. And this capping is nothing but it is 7-methyl 5-guanosine triphosphate. This form. 7-methyl 5-guanosine triphosphate capping is there on the 5 prime end, and on the 3 prime end there is a multiple number of adenine residues are there. So this is what is known as polyadenylation. Polyadenylation process is done. Polyadenylation, it is around something around 200 to 300 adenylate residues are added, which has been given in your NCERT book also. 200 to 300 adenylate residues are added at the 3 prime end, which is known as polyadenylation process. And these 3 processes, one is 5 prime capping. Actually, this guanosine residue which is added over is inverted guanosine. It's another technical thing. Just I added over here. So this is 5 prime capping, then 3 prime polyadenylation, and this splicing. And this splicing is done by an enzyme which is known as spliceosome. This is also known as SNRNP. SNRNP is basically it's a enzyme which basically cleaved these introns. This is a small nuclear protein plus small nuclear protein is there. It's a type of protein which basically do the splicing activity, and that is what is known as spliceosome. So this is the second additional feature which has been done. Second thing, which is not entirely here, because here the mRNA which has been formed is absolutely of mRNA, there is no any, absolutely of exons, there is no any introns is there. So directly protein is synthesizing. Now second thing, third point. Third point is here. The third point is the mRNA which has been formed. Now here I wrote it down. mRNA which has been formed, that is the process of transcription and translation is coupled. Process of transcription and translation is coupled. Now, if you ask me that what actually is the coupling, what do you mean by this coupling process? I am telling you, this is the transcription bubble. Transcription bubble. As you know, because in this particular video I am not elaborating what is transcription bubble, because in the next video I will upload the exactly position of this process of initiation. I will let you know the transcription bubble, because it is the same thing which has been happened as in case of DNA replication, where there is replication bubble is formed. In the similar fashion, here the transcription bubble is formed, and here the mRNA is formed. The nucleotide sequences, as you know, that available in all the double strands. Now here the mRNA is formed, 5 prime to 3 prime. That is the 5 prime to 3 prime mRNA is formed, already forming. On the one side mRNA is forming, and ribosome they are getting attached. Ribosomes are getting attached, and process of polypeptide chain is forming. Process of polypeptide chain is forming here. Polypeptide chains are forming over here, and the process of transcription is still not complete. The transcription and translation both is taking place simultaneously, which is known as transcription translation coupling or transcription protein synthesis coupling factor is taking place simultaneously in prokaryotic cell, which is not a characteristic feature of eukaryotic, because here the thing is like that mRNA which has been formed, it is coming through the nuclear core, and here in the cytoplasm the process of translation is taking place, which is a step, is a next step of the transcription. Here transcription and translation both are taking place simultaneously. So that is the third difference. Yes. What is the fourth one? Fourth one, I would like to say that there is a single RNA polymerase enzyme. There is a single RNA polymerase enzyme which is taking care of the entire process. Entire process of transcription is taken care by the RNA polymerase enzyme in case of prokaryotic cell. But in case of eukaryotic cell, this is RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. The three enzymes are taking care of the production. This RNA polymerase is a single enzyme which forms the rRNA, mRNA, and pRNA. All the three types of RNA is made by RNA polymerase enzyme, single RNA polymerase enzyme. But in this case, the three types of polymerases, this one, RNA polymerase 1 forms the R1, RNA polymerase 2 forms the mRNA. Now this mRNA, actually RNA polymerase 2 forms initially the hnRNA, which is the precursor of is the precursor of mRNA. This is the way the NCERT also elaborated things. RNA polymerase 3 forms the pRNA. See, these three different types of RNAs are involved here because the process of complexity is much more here in case of eukaryotic cell, while in case of prokaryotic cell it is much less complex. Okay, now the last and the most important point which I would like to write here, that here the RNA is polycistronic. Excellent. Polycistronic, and you can say the structural gene is also polycistronic. Polycistronic gene is there. In case of prokaryotic cell, polycistronic, that means a multiple gene. What is cistron? For that you have to understand what is cistron. Cistron is basically a structural unit of gene. Unit of gene, basically involved in, it is the smallest unit of gene basically involved in formation of polypeptide chain. Polypeptide chain. Okay. So here in this case is so many cistrons are involved, like this X, Y, Z. Different types of cisterns are there, and they are involved in the formation of different types of polypeptide chain. Multiple polypeptide chains are formed. Okay, here. But in case of eukaryotic cell, this is not polycistronic. This is monocistronic. Monocistronic gene is there. Single gene controls the single polypeptide chain. Here, single gene controls the single polypeptide chain. There are so many cisterns are there to take care of the entire protein synthesis. So this polycistronic, this is a single cistern is there in case of eukaryotic cell. So these are the basically these are the 5 characteristic differences. Although there may be much more differences you can say, but I am not going to. This is the major 5 differences. There may be some other because there are various aspects by which I can differentiate and I can draw the differences between the prokaryotic and eukaryotic cell transcription process, but that is not the major one. These are the major one. These 5 major differences are there between the prokaryotic cell and eukaryotic cell. I hope you will be benefited by this particular lecture. Thank you.